I want to, on the one hand, talk about a positive long-term outcome, namely, hopefully long-lived immunological memory, but then also a long-term outcome that has actually drawn our attention to not only COVID, but also other post-viral or virus-induced long-term issues, which we may call long COVID or post-acute COVID-19 syndrome. Now, in my talk, I'd like to start with looking at long-lived memory CD8 T cells. And there, what we try to do is identify single cells on a clonal level that are SARS-CoV-2 specific effector and memory cells. We characterized them in terms of their phenotype and their transcriptome using CytSeq, as also mentioned by the previous speaker, and looked then at short-lived versus long-lived CD8 T cells. And I would like to present our data and then discuss some implications for CD8 T cell immunity. In the second part, I'd like to present some also very recent data on post-acute COVID-19 syndrome, which I will refer to as by its acronym PAX, or also known as long COVID. And there we've done a study where we try to identify factors that correlate with increased risk of PAX, and including actually identification of an immunoglobulin signature that seems to correlate with increased risk of PAX. And I would like to then discuss with you the implications, potential implications for prevention, diagnosis, and treatment of PAX. Now, upon a challenge to our immune system, particularly with an acute infection, our immune system actually reacts by first mounting a very rapid innate immune response, as we have just heard from the previous speaker, which is then followed by a slightly delayed but very potent adaptive immune response. Ideally, as the adaptive immune response takes over and controls the virus, the innate immune response should wane and then die off. And the adaptive immune response then, once it has eliminated the insulting pathogens, such as, for example, a virus, it should then be reduced. But a small fraction of those adaptive immune cells, including T cells and B cells, should then survive as memory. Now, this is shown here graphically and schematically, and I would like to discuss this for pathogen-specific T cells, as this is relevant for the talk. Now, here again, those pathogen-specific T cells, they will, following the innate immune response, actually expand vigorously. Here in green are the virus-specific or pathogen-specific T cells that expand, that control the virus, and upon controlling the virus, they undergo apoptosis, which leads to a contraction of the T cell response, and then some T cells are then selected to become memory T cells. Now, previous work has suggested that different external determinants may actually guide T cells to either becoming short-lived effector cells or long-lived memory precursors, which then transit and become long-lived memory cells. I have listed here a few examples, such as strong TCR signaling, strong interleukin-2 signaling, or weak IL-7 signaling, leading rather to short-lived effector T cells, and the opposite, namely weak TCR signaling, weak IL-2 signaling, but strong IL-7 signaling, being correlated with memory T cell precursors. Now, in terms of T cell responses to SARS-CoV-2, we know very well that these are generated. Here are some very early data from the group of Shane Crotty and Alessandro Sette at the La Jolla Institute of Allergy and Immunology, showing clear and robust CD4 T cell responses and CD8 T cell responses to SARS-CoV-2 peptides. Now, these assays were done using an activation-induced marker assay here for the CD4s, OX40 and 41BB or CD137 were used, that were expressed actually on antigen-specific CD4 cells upon in vitro restimulation with those peptides versus CD69 and 41BB on CD8 T cells. Now, for our purposes, we couldn't use such an AIM assay because we wanted to actually detect CD8 T cells that are SARS-CoV-2 specific in an unperturbed state. Thus, we could actually then look at their transcriptome. So therefore, we used another approach. We used dextramers that were tagged with an oligo. The aims of our study were to assess antigen-specific CD8 T cells up to 1 year after infection in order to identify long-lived memory cells, to define phenotypical trajectories at the T cell population and clonal level, and to track actually those T cell clones that persisted or disappeared at the memory stage in order to try and identify features of long-lived memory cells. In order to do so, we set out and established a COVID-19 cohort here in Zurich in collaboration with 4 other hospitals, including the University Hospital Zurich. And we collected and deeply characterized 175 individuals already in 2020. So they got contact with the original Wuhan strain. And then we sampled them at the acute infection 6 months later and 1 year later, and also got clinical data as well as PBMCs and sera. Now, as I mentioned, we did not use an AIM assay, but we used SARS-CoV-2 specific MHC class 1 dextramers. And here is shown one example. Here, a healthy individual that does not show much background staining versus a patient at acute 6 months or 1 year showing a robust CoV-2 dex positive fraction. Now, I will refer to these CoV-2 dex positive cells as SARS-CoV-2 specific cells, but it is clear that even in the CoV-2 dex negative fraction, we will have cells that are SARS-CoV-2 specific, which simply did not recognize this dextramers. Now, doing so, we were clearly able to stain for CoV-2 dex positive CD8 T cells compared to the levels or the background levels in non-infected healthy individuals. You could see that there was a robust expansion of these CoV-2 dex positive cells at the acute phase, which was maintained at 6 months and at 1 year. Looking at their phenotype by using spectral flow cytometry, we could see that in comparison to the total CD8 T cells here shown in gray, the CoV-2 dex positive cells were actually gathered here in this corner, and they were CCR7 low, IL-7 receptor alpha low as expected, but some of those cells were highly proliferative as shown by Ki67 expression. This is also shown here that by showing that cells at acute infection were highly proliferative, but this proliferative behavior actually returned to background levels at 6 months and at 1 year. They also upregulated HLA-DR, which is a typical activation marker on human T cells, and they upregulated granzyme B, which stayed high actually up to 6 months before then returning to lower levels. They downregulated IL-7 receptor alpha, which was also accepted, and then gradually upregulated it over a year, which we thought was interesting. So it was not a very rapid reupregulation, but a gradual upregulation. Now, in terms of the transcriptome, again, comparing the entire CD8 T cell population, we could find, or we could identify 12 clusters, and some of these clusters actually were more predominant during acute infection, whereas others were more predominant at the memory phase. And this is also shown here. The red dots are COV2 DEX positive cells at the acute stage. They gathered here in these clusters, whereas COV2 DEX positive cells at the memory stage gathered rather here. Now, looking at the genes, we found that at the acute phase clusters 1, 2, and 12 actually were more predominant, which correlated actually with cytotoxicity, with activation, and with proliferation. Conversely, at 6 months, clusters 3, 6, and 11 were very predominant, which showed in cluster 11 actually a very strong interferon response and cytokine production. Now, looking at single COV2 specific CD8 T cells, we could actually look at them both at the acute phase in terms of epitopes as well as at 6 months, and we could also sequence the CDR3s of their TCRs and could thus actually observe cells that contracted in comparison, so from acute to 6 months, whereas others actually rather seem to expand from acute to 6 months. Now, of course, this is relative frequency and has to be, it does not say something about the actual clonal abundance. We also looked at single clones, and there again, we saw the same types of behavior. So the identical same clones actually at acute infection were rather in these clusters up here, whereas at the memory phase, those clones were then found in these clusters, clustering here and including the interferon and cytokine clusters. And that was also true for the other clones on a clonal level. And if we looked at them and looked at different gene signatures, then those acute gene signatures decreased actually, whereas the recovery gene signatures increased. And here is an example also on the genetic level of Ki67, which clearly decreased from acute to 6 months. And the same is true for HLA-DR. Phenotypically, we used the classical designations or differentiation into either central memory, which are CCR7 high, CD45RA low, effector memory, which are low for RA, 45RA and CCR7, and CD45RA positive effector memory, abbreviated as TEMRA. And then these cells here be further subdivided based on CD95 into stem cell memory and naive cells. And here we see COV2 DEX negative CD8 T cells, so the bulk of CD8 T cells, whereas here we have the COV2 DEX positive ones. And what is very interesting is that at the acute stage, as expected, we see mostly effector and effector memory cells, but then those cells actually gradually become TEMRA cells. And this is also shown here in summary. So the predominant phenotype present actually at 6 and 12 months was clearly TEMRA, and stem cell memory actually only represented a small fraction of those long persisting cells. These cells also gradually upregulated TCF1 transcription factor and downregulated TBET. Now, another interesting observation we made is when we looked at those patients that had mild disease versus severe disease, then the severe disease patients at 1 year after infection had actually a higher fraction of TEMRA, even though mild disease patients also had the predominant features of TEMRA. So this might indicate that accompanying inflammation might be a driver of TEMRA. Now, looking at single clones, we then looked at those genes at the acute stage in the clones that then went on and persisted into long-lived memory cells versus those clones that actually disappeared, i.e. non-persisters. And what we could see is that a strong interferon gamma, interferon alpha, and TNF signaling via NF-kappa B were enriched in persistent, whereas in non-persisters, a strong mTORC and in proliferation associated signals were present. This is also shown here that up here in persisters, we have interferon related or inducible genes and CD45RA, which speaks again for TEMRA, whereas here in the non-persisters, we have proliferation. So in summary, we identified single clones of SARS-CoV-2 specific long-lived memory CD8 T cells up to 1 year. TEMRA was the predominant phenotype, which was more seen in severe disease than in mild disease, which contrasts with previous reports, for example, using yellow fever virus vaccination in humans that found T stem cell memory. I'm happy to discuss that later. And the transcriptome signature of interferon signaling and cytokine production characterized those SARS-CoV-2 specific effector cells that became long-lived memory. Now, the second story I'd like to share with you is also a very recent story from our department where we looked at PAX or long COVID. And PAX, at the time we started looking at it, which was at the end of 2020, was defined as symptoms that persisted for 4 weeks and longer. Because at around 3 to early 4 weeks, it is believed that virus cannot be isolated anymore from the respiratory tract. So it is expected that symptoms should actually then be gone. And if they persist, then this is then called PAX. And the longer they persist, then they can become actually post-COVID-19 syndrome. Now, what is PAX and what symptoms are most often seen? So the patients we see often have fatigue, some have cough and dyspnea, some have actually what is referred to as brain fog, which are cognitive impairment, chest tightness, palpitations, and memory issues are other symptoms, typical symptoms of PAX. So what we did is we again took our COVID-19 cohort of 175 individuals, and in parallel, we had a control group that we included at the very same time in order to correct actually or observe factors that are not due to SARS-CoV-2 infection, but to the pandemic-related environmental changes, such as working in home office, having, let's say, more stress at work or with work or at home. And this control group actually did not have any clinical signs of COVID, nor were they serologically or PCR positive for SARS-CoV-2. We took medical history and laboratory testing, and then did the same at 6 months and at 1 year. And then we took a separate validation cohort of 395 patients with confirmed COVID-19. Now, if we look at this cohort, then we had about 90 patients with mild COVID and about 45 with severe. If you look at the general population, then very clearly the fraction of severe patients here is clearly larger than what we see in the general population. But this was on purpose in order to have also an insight into long COVID in those that get severe COVID, and also in those that get hospitalized, and not only in outpatients. As mentioned, we had a control cohort of 40 individuals and a validation cohort, and we looked at any prolonged symptoms to define that persisted actually for 28 days longer to define long COVID. Now, unlike a study we had done earlier where we looked at SARS-CoV-2 specific antibodies, both IgA and IgG, both in the blood as well as in mucosal fluids such as nasal secretions and tear fluid and saliva. Here in this study, in the current study, we looked at total immunoglobulins. So not only the SARS-CoV-2 specific, but really the total levels. And what we found was very interesting. So IgM, total IgM, was lower actually in patients with severe COVID compared to mild COVID or healthy controls. And if we looked at those that did not have PAX here in orange and those that did have PAX here in red, then those that had PAX had relatively but significantly lower levels of IgM. Now this contrasted actually with IgA, and it also contrasted with IgG1, but in terms of IgG3, we again saw a similar trend in that those with PAX had actually lower levels of IgG3. Now, this allowed us to make an interaction function of IgG3 and IgM, in that if IgG3 or IgM was low, then the predicted probability of PAX was very high. And this was also true even before infection, as we saw in 5 patients that were previously healthy and then contracted PAX, COVID and then PAX. Now, also patient age, a higher patient age and the number of symptoms actually correlated with increased risk. And this in total then allowed us, together with a history of asthma, to make a predictive score that actually performed quite decent, both during primary infection as well as at 6 months. and also in our validation cohort, where it performed well both in the outpatient, but especially well in the hospitalized patients. So what this could be used for is actually to group patients into very high, and moderate risk patients. Now, with this, I hope to have shown you that we found age, history of asthma, symptoms during acute COVID, and this immunoglobulin signature as important risk factors of long COVID. Now, I'd like to acknowledge the people that did the work from my work, especially Sara Adamo for the first study and Carlo Cervia for the second study, as well as our collaborators and our funders. And thank you very much for your attention.
Thank you very much, Dr. Boyman. Let's see for questions from the attendees. Okay, I see a question from Eric.
Hi, Dr. Boyman. Thank you for the talk. I have two questions. The first is there any evidence in these patients from the PAC study whether they have maybe a hidden reservoir of virus in the body that's somehow affecting outcomes? So, for example, we've heard talks in the past indicating that there are reservoirs of virus in the gut that persist for quite a long time. And the second question is related to the differences in the IgM level, for example. Do you have any sense of why there's a difference in these patients?
Yeah, thank you very much for these questions. So to the first question about hidden viral reservoirs, we think that that is a possibility, but we think that this actually would only explain a certain fraction of long COVID patients. So we clearly have indications that there are different, let's say, mechanisms that lead to long COVID. So if I may actually go back one slide, we think that what is important and what should come out of these studies is a subgrouping of PACs based on proposed causes, and one cause could be a relative immunodeficiency, and this could actually correlate potentially with hidden virus reservoirs, for example, in the gut. But there are clearly indications also for protracted inflammation being potentially a cause of long COVID, or actually autoimmunity. So I would actually tend towards saying hidden viral reservoirs may be a reason, but I don't think that those are the predominant reason, but that there are rather a dysregulated inflammation, autoimmunity, or other causes that lead to that. As to your first question, why do some patients have lower IgM? I think it's sort of there. So these low IgM and/or low IgG3 levels, we think really tell something about how their immune system generally reacts. So we made an interesting observation that those patients that have a history of asthma, they had relatively lower levels of IgG3. So it might really be that their type 2 immune skewing might actually play a role in rather responding not with IgG3. And as for IgM, we saw that individuals that are rather older have actually slightly lower levels of IgM. So this might also then correlate with older age and hence more severe COVID, and therefore a risk for long COVID.